how looking at cellular aging and animal, especially mouse aging, might actually help us not only to understand human aging, but in fact to modify it. And the first question that actually raises here is, why should we increase healthy human lifespan? If you look at these data, you see that human average life expectancy has been increasing at an truly astonishing rate over the last almost 200 years. That's as far as we really have reliable data on it. And it has been increasing on a linear rate with so far really no indication of slowdown. And in fact, predictions of a maximum human age, as you can see all over there, starting from the 1920s, have all been too low. And the actual rise of human life expectancy was faster than all those. So that is, of course, good news in the first place, because if life expectancy increases by something like 1 year every 3 to 4 calendar years, then for you, after you have listened to this talk, your life expectancy has also increased by something like 15 minutes. Very nice. However, that's not the whole story. And that's, I would like to show you in the next 2 slides. So here you see basically the same data as before, a little bit more recent, specific to the European Union. So you have here the means and the ranges of life expectancy in the EU in 2005 in gray and in 2010 in yellow. And this is separated into the old EU states, that's the EU 15, and in the newer ones, those that became members of the EU in 2004, and it's separated into men and women. And of course, as we know, women live longer, and people who are less well off live shorter, and that is why we see quite a significant difference between life expectancies in the old EU states and in the newer ones. But as I said before, within these 5 years, from 2005 to 2010, life expectancy, both for men, both for women, in all these different countries increased by about between 1 and 2 years. So the trend's still going on. But now is the problem. That is life expectancy. Healthy life expectancy is different. Of course, it's shorter. Now you have what's called health span. So the age at which the first serious chronic condition starts. And what you see here is, first place, differences between men and women are less marked now. Second, of course, variation is larger. And third, and importantly, if there is an increase between 2005 and 2010 in healthy life expectancy, it's significantly smaller than that of life expectancy. And in some cases, especially in the new member states, there's actually no increase in healthy life expectancy. So what we have is a situation where lifespan increases, but health span does not, or increases more slowly. That's something which I like to call the longevity trap, which means that modern medicine is very good in managing age-associated chronic diseases, making them, if you like, more bearable, allowing you to live with them. But in the end, that results in an increasingly longer period of diminished health during the end of life, which means costs for society and, of course, costs for the individual. In other words, we do live longer now, but we still age at the same rate, more or less. I'll give you an example. And I'm talking about long-term survivors of tumors, of cancer. And that's an important issue these days because for many, many cancers, with some very notable exceptions, treatment success rate is pretty close to 100%. So you don't die anymore from most cancers. That's great news. But that means that alone in the UK, there is something between 1 and 2 million people who are long-term cancer survivors. And these people do show a wide range of diseases. They show increased mortality. So the standardized mortality rate is, depending on the study, between 2 and 8 times higher than in the general population. They show multimorbidity. And the diseases that they suffer from are secondary tumors, that is, tumors not related to the primary tumors. That's cardiovascular disease, that's diabetes, osteoporosis, cognitive decline, fatigue, frailty. In fact, what they show is they show premature aging. And we do know that this is not in the most cases, not caused by the primary disease, but by the treatment. And in addition, there is nothing except some lifestyle counseling that we can offer those people for these increased morbidity and mortality. So I think the question, how do we understand aging and what can we do to delay or postpone or maybe even cure it, is a very important one. And it's probably worth stopping at that point for a second and thinking back just a couple of years, because if you allow me a personal word, when I started in the field of aging in 1980, 40 years ago, that was a time when many of my peers actually said, what do you want to do? Why do you want to study aging? You don't study aging, you experience it, and it's just too complicated, and you can't do anything about it anyway. That's just 40 years ago. So we have come a long, long way from that. And I'll give you some examples. So we've learned that there are a number of pretty clear processes, hallmarks of aging, as Carlos Lopez-Otin called them in 2013, that are defining how we age. And the next slide shows the same hallmarks in a bit more of a causal sequence. So Carlos discriminated between 3 groups. One is the primary hallmarks: genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, which are causes of damage that drive aging. Then he saw hallmarks that he called antagonistic because they are responses to this primary damage and as such adaptive, but at the same time, while they are short-term advantageous for the system, for the animal, for the human, they will in the long term contribute to the aging process. And that's mitochondrial dysfunction, cell senescence, and deregulation of nutrient sensing. And then there are also hallmarks that are integrative, systemic, like stem cell exhaustion and altered communication, and those together drive the aging process. Now, I will focus on one of these here in the rest of the talk, and that's cellular senescence. And I do that, well, of course, because I know most about it, more than about the other things, but also because it really has very, very strong links, and I would say a sort of integrative function for all the other hallmarks. So all of the primary ones are drivers of cell senescence. Mitochondrial dysfunction and deregulated nutrient sensing are parts of the phenotype. And we still don't know how much cell senescence is not only a consequence, but actually also a cause of those, as it is a cause of stem cell exhaustion and the cause of altered cell-cell communications. So I'll focus on that. And that's a story that begins with this guy, Len Hayflick, shown here in 2004 when he was our guest of honor when we started our center in Newcastle. And he, in 1961, not just made a remarkable experiment, but also understood remarkably well the experiment he did. So what he did was pretty simple. He isolated primary human cells from embryos in those days and cultivated them. And they, of course, started to divide and divided and divided and divided. And you could quantify these divisions by counting the population doublings of the cell numbers. And then he found that after a couple of months, the division of these cells would stop. And in his cases, in his cells and under his conditions, that was at about 50 population doublings. And as you can see in this slide, cells also look different. And they would never start to divide again. They would still be alive for a year or two, look more and more strange and unhealthy, but they would not divide anymore. And the remarkable thing about Len Hayflick was that he recognized this was not just because he wasn't able to cultivate cells well. It was not a cell culture artifact. It was an inherent property of these cells. These, what we call now the Hayflick limit, this replicative senescence was an actual property of dividing human cells, primary cells. And it took 30 years, almost 30 years, for people to understand the clock that was behind that. And that clock was then found to be in the telomeres. So telomeres are shown here on this human metaphase as the ends of all chromosomes indicated by a fluorescent label here on the tips of the chromosomes. And Calvin Harley in 1990 published this paper where he showed that in the same type of primary human cells, those telomeres shortened with increasing number of population doublings as indicated here on the top. So in this cell line that made more than the 50 population doublings Len Hayflick's line did, went up to almost 90, but principle was the same. Telomere shortening with increasing number of population doublings. So there was something like a clock. And that was in fact not just a clock counting time, but a clock counting biological time in terms of stress exposure is something we then found. So, and the next important step was that people were able to show that these telomere shortening at some stage would induce what then became known as a DNA damage response. So Cells recognize DNA damage by forming foci, protein foci comprised of many different proteins, including this one here, a phosphorylated form, gamma, of the histone H2AX. And in 2003, it was found that in the senescent cells, these foci in green would co-localize with telomeres to a large extent. And that was done with different technologies. So short telomeres would induce a DNA damage response. And that led to a very simple and still valid hypothesis, namely that during cell division, telomeres would shorten, so became uncapped. That is recognized by these DNA damage proteins, which then in turn would activate cell cycle proteins, including P21, P16, P53, and that would lead to cell cycle arrest and replicative senescence. Now, one thing to say here is that cell senescence is much more than just the proliferation arrest. And that's shown immediately if you look at those cells. Was shown in the Hayflick cells I showed you before. This is a more recent version of that. You see senescent cells look different. They are larger, they have expanded nuclei, nuclear shape is different. And what you also see in this slide is the mitochondrial network looks very different. So we stain here mitochondria with both a green and a red dye. The green one stains all mitochondria. The red one stains only mitochondria with a high membrane potential, so actively respiring mitochondria. And you see that we lose a lot of the red in the transition from young to senescent cells. So mitochondrial dysfunction is one of these parts of the senescent phenotype, and there are others. So these are a sort of minimum number of senescence building blocks and phenotypes. So you have a DNA damage response that drives the cell cycle arrest. You have activation of frequently pro-inflammatory transcription factors like NF-kappa B, CEPB, others that drive the core of what's been called the SASP or senescence-associated secretory phenotype. So many of these factors that are activated by these transcription factors, like interleukins, are part of the are secreted from senescent cells. I mentioned already mitochondrial dysfunction, which drives enhanced production of reactive oxygen species in senescence. Nutrient signaling, dysregulated nutrient signaling is part of the senescent phenotype, and that is maintained via a different responsibility to the mTOR. Autophagy and mitophagy is dysfunctional. That leads to one of the typical characteristics of senescent cells, that they are highly expressing beta-galactosidase and other lysosomal enzymes. And many of these processes are due to epigenetic reprogramming, which is both on the messenger and on the microRNA level. And it's also important to say that all these building blocks and phenotypes are closely interrelated in regulatory networks. And I don't want to go into details here. Mitochondrial nuclear crosstalk plays an important role, and others. So, and one of the things that had been discussed a lot in the field is Okay, so Len found the senescence phenotype. It's true, but it's in vitro, and it might well be an in vitro cell culture artifact. That's been discussed until something like 10, 12 years ago. And a couple of people, including us, however, showed that senescent cells, in fact, are part of the normal aging process. They accumulate with aging in vivo. First paper came already in the 1990s from Judith Campisi's group, and we published a couple of years back a paper that showed that, in fact, senescent cells not only accumulate with age, the rate of accumulation predicts lifespan. So what I'm showing you here is data from 5 different mouse strains with very different lifespans. So here on this graph, you see maximum life expectancy in those mouse strains. Some have very short lives. Green one, that is a telomerase knockout mouse. Others have very long life. This here is the black one is a C56 standard mouse strain, which is used in many labs. And if you put that one under dietary restriction, you get a very significant increase in lifespan. Now, if you measure senescent cell frequencies, in this case in the liver, using a number of different markers here, those 5 graphs are all the same thing, frequencies of senescent cells in liver using different markers, then we see remarkably a pretty linear increase over relative lifespan, percentage of maximum lifespan completed, which basically means that in mice that have short lifespan, senescent cells accumulate faster, and in mice that have long lifespan, senescent cells accumulate more slowly, and that's shown in the 2 lower graphs here, both for the liver and for the gut, especially the crypt enterocytes. So in these, at least in these 2 tissues where we looked, the accumulation rate of senescent cells predicts pretty nicely lifespan of the mouse strain. Okay. So, but why do senescent cells accumulate? There's at least 2 possibilities here. Of course, they are generated by stress. That could be replication stress. There's also oxidative damage. That could be, like in the case of the tumor survivors, stress induced by treatments, whether radiation or chemotherapy. That all leads to accumulation of senescent cells. On the other side, the immune system is actually pretty good in detecting those cells and in killing them. So we have an equilibrium between the 2 processes. However, one of the problems is the immune system, the quality of it decreases during aging. So the immune surveillance will decrease. And that, of course, is one possibility that will lead to accumulation of senescent cells. There's also another one, and that is senescent cells actually induce senescence themselves in their environment. So if you like, senescent cells are something which you could call infectious, and that shifts the equilibrium, and then even uncompromised immune surveillance might not be sufficient to keep their numbers in check. And I'll show you some data on that. Here, this is showing some of the building blocks of senescence in a network structure. The point I want to make here is that many of the factors that are overproduced in senescent cells are actually secreted from them. That is the case for NF-kappa B-driven factors, interleukins like IL-6, IL-8. That is the case for reactive oxygen species. And for others. And here's a list of some of the factors that together make up the senescence-associated secretory phenotype: interleukins, chemokines, growth factors, TGF-beta, reactive oxygen species, microRNAs and proteins, also oxidized lipids, many of these in exosomes. And all these are highly bioactive molecules. So they have impacts on their environment. And the first one has been published already almost 20 years ago by showing that senescent cells do have a pro-oncogenic effect on their environment. So the experiment was done by the Campisi group, and what they did was they xenotransplanted different human tumor cells, HACATs, SCP, these MDA231, into nude mice. And they transplanted low numbers. And then they looked at the development of tumors, measuring tumor size in the flanks of those mice. And because those tumor cells were only few, they did not really give rise to tumors during the observation period. That's here in the first row. The second experiment was the same one, but in addition to the tumor cells, they also injected non-senescent or pre-senescent fibroblasts. And again, growth of the tumor cells was very, very limited. However, when they did the final experiment, injected same number of tumor cells and then a number of senescent cells with them, suddenly tumors grew. So those tumors, they are not from the senescent cells. The senescent cells can't form tumors, but the senescent cells can stimulate tumor growth. So they do have significant effects on their environment. And that is, so on tumor cells, which have broken down checkpoint proteins, they induce growth. On normal cells, they induce senescence. So this is an experiment where we grew a senescent cell, that's the one labeled in red here, big one, together with young cells. And you see the nuclei of 5 young cells in the vicinity of the senescent cell. And we put them together for a couple of days, and then we checked for one marker of senescence, namely the co-localization of this DNA damage protein, gamma HJX in green, and of PML bodies. And you see the nucleus of the senescent cell, and co-localization is shown in white here. The nucleus of the senescent cell is full of those. But also the nuclei of those cells that 10 days ago were very, very young and healthy started to show these co-localization, indicating that the senescent cell here induced senescence markers in their environment. And that happens also in vivo. So when we inject senescent fibroblasts into mice, either subcutaneously or in the muscle, and as control use non-senescent cells, then those cells were labeled fluorescently and with luciferase, so we could find the injected cells after a few weeks here in the liver, in the muscle, and here under the skin. What we then found was that in the vicinity to those injected senescent cells, the original mouse cells showed more and more senescence different markers. So also in vivo, senescent cells induce senescence in their environment. And that has functional consequences. That's an experiment done by the group of Jim Kirkland at Mayo Clinic. Basically the same experiment. They injected either non-senescent control cells or senescent cells into young mice in the fat tissue in this case, waited for a while, and then performed functional measurements. And they waited for at least a month. After that time, the injected cells were pretty much gone, but they found enhanced markers of senescence in the mouse tissues. And importantly, they then checked essentially muscle function in those mice by the ability to run on a rotor rod, by the ability to hang on a wire, and by grip strength. And in each case, they found a decrease of these functional parameters with increasing numbers of injected senescent cells. And again, those cells had not been injected into muscle, they had been injected into fat. So it's mediated by systemic effects of these senescent cells. So senescent cells are, in aging at least, a bad thing. One thing I should also mention, that is, senescence is not limited to proliferation competent cells. When a couple of years we looked for senescence markers in different tissues in mice, and were originally quite surprised to find an accumulation of those markers in postmitotic cells, specifically in neurons. So here on the left, shown in Purkinje cells, in old mice, 32 months old, green is always the senescence marker, and you see the accumulation. These are the Purkinje cells here. See the accumulation of the green in those cells, different markers, multiple markers in the same cell. Here, senescence-associated Zen-beta-gal staining, again at these cells, and also in peripheral mesenteric neurons. So that's markers of senescence accumulating with age. Now, is this really senescence? So I showed you that slide before that defines senescence as a primarily telomere damage mediated activation of checkpoint factors, like P21, P16. So we tested that in a transgenic model. Again, we were looking at the brain, and here those cells again are the Purkinje cells around here in essentially wild-type mice. And then in mice where there was a knockout of one of the telomerase proteins, leading in later generations to short dysfunctional telomeres, and brown always indicates the marker. So again, we find senescence markers in these cells in the mice with late generation telomerase knockout. Then now we also knocked out the gene for P21, CDKN1A, in these mice, we lost the induction of senescence in those cells, proving that yes, it was telomere mediated and depending on signaling through P21. So truly a senescence process. And by now we know that essentially in all types of postmitotic cells that have been looked at, cardiomyocytes, myofibers, other types of senescent cells, other types of postmitotic cells, we find markers of senescence accumulating. So now finally coming to intervention. Yes, killing senescent cells is an anti-aging intervention. And I still remember it was discussed for the first time in 2007. Jim Kirkland had this at Mayo had this idea, and when he discussed that with me, I said, "Well, I don't believe it. First of all, it will be technically extremely complicated to specifically kill senescent cells. And second, it must simply be so that it must be better to have a senescent cell than no cell. Well, I was wrong on both counts. It turned out to be possible, quite a ingenious system where senescence, where transgenic mice have been generated, where the full-length promoter of P16 drives a caspase. So if P16 is transcriptionally activated, caspase is expressed. This caspase is linked to an FKBP binding protein, which anchors it in the membrane. And this protein can be dimerized by a drug, AP20187. So only if the mice get the drug, the FKBPs are dimerized, and the caspases are able to activate each other, and then they drive apoptosis. So senescent cells where P16 is transcriptionally activated will be killed when the mice are fed with the drug. And that improves their aging process. It enhances lifespan. Mice that have been fed the drug have better fur, they don't show kyphosis and other aging markers. So that's the proof of principle. Then people have been able to find drugs that can be fed to non-genetically modified mice by doing interactome searches and then identifying these drugs. I think I'll not go into details like that. There are still searches are ongoing for better drugs. In this case, that is one which seems to work very well, at least in vitro, where in a co-culture of senescent cells, green labeled, and young cells, only the young cells remain after a couple of days, and they are still able to proliferate while all the greens are gone, and you see quite a nice therapeutic window for this drug. So yes, there are now drugs that can be fed to cells, probably to mice, and that in fact do work. So this shows the same data as before. Injection of senescent cells leads to functional deterioration of muscle function, but if we treat those mice where the senescent cells were injected with a combination of the first generation senolytics, we improve all those functions. And there's now quite a literature on successful treatment or at least postponement of multiple age-associated conditions in mice by different senolytic approaches. Again, I don't want to go into details here. And there's also reports from first 2 or 3 clinical trials in men with senolytics, again, in generally encouraging. I would like to finish off in 3 minutes. I would like to go back on to the tumor survivor story. I showed you that slide before. In order to test whether it would be possible to treat these accelerated aging conditions in tumor survivors, we performed a preclinical experiment in mice where we irradiated young mice, waited until the acute radiation sickness was abated, and then followed those mice for another year and saw that they showed premature aging. And that was, of course, driven by the irradiation-induced premature senescence, inducing bystander senescence. So we reasoned that if you would treat mice at that early stage with a senolytic, we would kill the senescent cells and we should improve outcome. And we check different parameters. One of them is frailty. Frailty index is a combination of a couple of easily to assess parameters. And you see here the increase of this frailty index in normally aging mice over 2 years. And you see also that after irradiation, in the red example, the frailty index increases about twice as fast in these mice. So what if we now treat the mice and we use 2 different senolytics and both of them worked? They do not stop the increase in the frailty index, but they reduce the rate of increase to one which is pretty much the same as in the wild type mice. In addition, it improves neuromuscular coordination, and it improves cognition, or better to say, short-term memory, as shown here by spontaneous alternation in a Y maze. It also improves or tends to improve the induction of tumors. Also, these experiments were not powered sufficiently well to show significance here. Final question that we asked was, what about normal tumor survivors? They will have already established premature frailty. Can we still improve their condition? So we wait in the mouse experiment until frailty is enhanced and then treat. And again, we reduce the further increase in frailty. However, effects on neuromuscular coordination are not significant, but we still get an improvement of short-term memory at the end of the observation period. And tumors, this treatment was too late to improve tumor incidence. So, in summary, yes, we can, right now, we can slow down at least premature aging in mice. There's no reason to assume that we would not be able to slow down or maybe even rescue premature aging in humans. We are still some way off from reversing aging, as in this nice picture. And whether we will ever be able to do that is a very different question. But I would say there is highly significant hope, even in the lifespan of a rather old guy like myself, that we would be able to slow down further aging. And with that, I stop sharing and invite you to ask questions.
Thank you so much, Thomas. Thank you for really this inspiring talk. So I'm quite sure that our students will raise their questions. So I really invite you. I also invite those that are following this talk by YouTube to put their questions on the chat. I need to I must apologize because we had an error in the link to the video that finally was redirected to the new one. So we have some people attending via YouTube. So please raise your questions in the chat. So our students now, the floor is yours to raise your questions to our really fantastic speaker, Thomas von Zglinicki. Someone wants to start?
I have a question, if I can.
Sure, Susanna.
Do we have some methods to assess the oxidative stress in humans?
To assess what in humans, please?
Oxidative stress.
Oh, yes, there are methods to do that in humans, with limitations. So let's see, people use methods to measure protein oxidation, lipid oxidation. There's a number. The typical issues are that you get a moment, a very limited picture of stress levels at that moment. So you have to be very, very careful to standardize your sampling. In fact, that was one of the reasons why we thought we would use telomere length as a measure of long-term cumulative oxidative stress, because telomere shortening is driven by long-term stress. But again, there's many factors that drive that. So I would probably need to think a moment about what I would prefer as the best method to measure oxidative stress. And if you really want to do that, you would need to think about, do you want to have cumulative measure? Do you want to have short-term indications? And you would need to use a couple of different methods.
So,
okay,
yes,
thank you.
Thank you. More questions?
Yes, can I ask a question?
Yes, Zglinicki, please.
So thank you for your talk. I was wondering, when you see the effects of removal of senescent cells on the brain, is that due to the removal of the SASP from the periphery that goes into the brain, or the removal of the senescent cells directly in the brain?
Yeah. Good question. Well, as always, a good question is one cannot really answer yet. That's because so far we don't have efficient techniques to remove senescent cell from one tissue, one organ, one cell type specifically. We have some data in the cell metabolism paper. We have some data which suggest that it's not just the removal of the SASP. So if we try to modify those SASP factors which come up predominantly in this senescence removal experiment there, then we can't reproduce the beneficial effect of the senescent cell removal. So we think that a couple of things. One is SASP factors influence each other in a very significant way. So probably it's will be very hard and sometimes work and sometimes not if you try to phenocopy a senescence removal by removal of a single SASP factor. The second thing is I'm pretty sure that we still don't know some of the relevant SASP factors. So just cytokines, interleukins is probably by far not the whole story. And finally, we see effects of senescent cells on cellular regeneration. So if we remove senescent cells, we seem to improve in different settings regenerative capability, not just that that's limited by senescence of the stem cells or progenitor cells itself. So seem to be secondary effects. So in short, we will have to wait until we really have techniques where we can remove specific types of senescent cells. And then we'll probably know more.
Okay, thank you.
Thank you, Ninke. Other questions?
I have one.
Yes, Inês.
Thank you for your presentation, Thomas. It was very great. So regarding this analytics approaches, I also believe they are a very nice tool to delete these senescent cells. But my question is, if we deliver these senolytic therapeutics as a prophylaxis treatment, do you believe this is feasible to humans? Because, for instance, navitoclax has some side effects. So I'm not sure if this is a good or a viable option. I just want to know what is your opinion about it.
Yes, I agree. It will depend on many things. First is specific situation of the humans under consideration. So if you want to treat, let's say, general aging, you would, there's no way you could use any of the present senolytics for that. If you look at people that are at the advanced stage, let's say, of frailty, and we know that frailty is a pretty good predictor of lifespan, of further degenerative events, things might look very different. If you look, let's say, at a population of people that are frail and that are needing major surgery, hip surgery or things like that, it might make sense, even with the present senolytics, to give them a short course and to improve their outcome. This is also why in those mice experiments I showed you, we actually treated those mice only for a period of 2 weeks, and then they were never treated again over the next year. And we followed them up for long term. The other thing to say in that context is, of course, already now we do have better stuff that is about as efficient as navitoclax, for instance, or D plus Q. Senostatics like metformin, we did the same experiment in those mice with metformin, treating them a little bit longer, but not for lifetime. And again, it worked. So there are possibilities, and I'm sure that second or third generation senolytics will also help to solve this situation.
Thank you, Thomas.
More questions? No more questions from you? Actually, I just have in the chat a message from Anabela Marisa Azul, just saying it's a really inspiring talk, and thank you so much, Thomas. So I just have a very short question, Thomas. So concerning these mouse experiments where there is a slight increase in cognitive performance, did actually people look into especially the hippocampus, hippocampal structure, to investigate whether or not these, I would say, the removal of senescent cells in the hippocampus could actually have some correlation with this better cognitive performance?
Yes. So in those experiments I showed you, we are doing that right now. I will actually have a lab meeting in an hour where I expect to see the first data of that. And we've had a paper this year in Aging Cell where we looked at the effect of senolytics in old brains, including hippocampus, and saw both improvements in memory and reduction of senescence markers. Yes.
Thank you, Thomas. So if you students, if you don't have more questions, and I just challenge you.
Wait, I have one extra question
Yes,
related to your question, because first, maybe stupid question, but does neuron get senescence or not?
Yes.
Yes,
yes, they do.
Okay.
And as I tried to show you, maybe I was too fast on that. To the best of our knowledge, we can say yes. And the best of our knowledge is paper which we published in 2012, showing accumulation of senescence markers. And we looked, I think, at 7 or so different senescence markers in neurons specifically, with age in mice. And then there have been 2 or 3 papers from other groups that also showed similar things. And we had a review paper this year where we summarized those data. So yes, neurons do get senescence, as do other postmitotic cells.
And do you think senolytics helps the cognitive functions because they work for this, they reduce the senescence of the neurons or other cells?
We think senolytics improve cognitive function. For that, we have evidence.
Okay, but you don't know which
that
cells.
is because they reduce frequencies of senescent neurons. That's much more complicated. What we know is that they improve in the mouse neurogenesis. But that's as far as I would be prepared to go.
Okay, thank you.
Welcome.
Thank you. So more questions? If not, so we will close the session now. So I really want to really give really warm thanks to Thomas for accepting this invitation. It was, yes, thank you, Takuya. I believe it is really worth.
Thank you. Thank you, and
Thomas.
have fun with the meeting.
Thank you, Thomas.
Bye.
Have a nice day. Bye-bye.
You too.
So for